Good afternoon, everybody. We are quite keen to start now, so if I could have your attention, please. I would like to introduce this session. I'm Adam Roberts, reader here in antimicrobial chemotherapy and resistance at LSTM, and I'm going to be chairing this session. My co-chair, fielding the online questions, is Dr. Fabrice Graf at the back. Hi, Fabrice. This session is all about resistance. Resistance is a strategic priority area of research for the school, as it is for many other societies and institutions, both academic and industrial. We are establishing quite a strong portfolio of research here at the school with resistance not only to antibacterials, but also to antifungals, antiprotozoans, antiparasitics, and also resistance in various vectors of disease. And we'll hear about aspects of all of these during the next 5 talks. The establishment of this strong portfolio of research is evidenced, I think, by some recent grants that we've got, which I just want to highlight. These are all in the last 12 months or so. We got about €1 million from the Joint Programme Initiative on AMR for the DECODE project, led by Fabrice Graf, to look at laboratory-based isolation of resistant bacteria from worldwide samples. We've recently had 2 MRC-funded projects worth approximately £1.5 million looking at resistance in disease vectors, led by Tony Nolan. We've received just over £2 million of Fleming Fund-funded work for a project led by Sally Theobald called the Gear Up Project, looking at gender considerations across the entire spectrum of the Fleming Fund portfolio of research, both here and within other institutions across many different countries. And finally, another one to highlight is a £3 million NIHR-funded OCT-AMR project led by Joe Valadez, looking at antibiotic usage and resistance in multiple countries around Africa. So these are just 4 of multiple grants that we've got. There are many others in negotiation, and I hope that just illustrates the strong basis we've got for research, all the way from laboratory-based science to policy implementation as well. So the whole spectrum of research along the resistance portfolio. So it's without any further ado that it's my pleasure to invite our first speaker, Dr. Catherine Moore, who's a reader at St George's University in London, and she's going to talk to us about... Well, I'm going to let you introduce yourself to me. Yes, I know.
I hope I don't break those. I haven't, so yes. There we go. There you go, Adam. I'm going to be talking about AMR and how it will remain the biggest global threat unless we change our practices. Most of what I'm going to talk about is on the global level, and it is actually WHO level, it is global burden of disease level, but I will take you very much closer to the coalface, I guess, in the last couple of slides and some of the work that I'm involved with. So, to start with, what are the drivers of AMR? Do we know what those drivers are? Are we sure we know what those drivers are? Well, what we've been talking about over the last few years is about the One Health notion. Now, if I can find the red marker, there we go. The One Health notion. So it's not just humans, but it's the environment in which we live in, it's animals, it's the whole gambit. And we should be thinking in that whole picture. But actually, then we come down to, well, most of the drivers that we have are due to poverty in humans. And we've talked, there's been a lot of talk about poverty and about health systems today. And a lot of what we talk about is that antibiotics and the overuse or the misuse of antibiotics drives antimicrobial resistance. And we can see that in the next couple of pictures. This one, so antibiotic resistance, is socioeconomic, it's antibiotic consumption, it's about infectious diseases. And then when we look at antibiotic use compared to antibiotic resistance, they are very much linked. But then often what we think about is antibiotic use in hospitals, because that's the data that we have. I will bring you back from the hospitals a bit to the community in the last couple of slides, because actually I think that what we have in the hospital may well be really resistant isolates, On the other hand, they may be the tip of the iceberg. So it may be that we have resistance everywhere and we're just not detecting it. So why do I say that AMR will remain the biggest global health threat? What evidence do I have to say that? Well, part of the evidence came from the GRAM project, which I led in the first round. And we published a paper in 2019 where we said between 1.3 and 5 million people died in 2019 in one single year because of AMR. There's been an update since then where we've said that actually those deaths have gone up because of methicillin-resistant Staph aureus, and there's more resistance to carbapenems over the last few years. And then actually, by 2050, more people will die because of antimicrobial resistance. And although we normally think that most of the people who die are under 5 years of age, with our increasing numbers of older people, they're more at risk as well. And so we'll see a much higher burden in older people. And so the paper that was published a couple of months ago said that AMR mortality is predicted to increase by 70% by 2050, and that we need improved access to antibiotics. But that is if we don't misuse those antibiotics and if we use them properly. And how can we start to track what's happening with antibiotic use and antibiotic resistance? Well, we've got the WHO track system, which looks at the national action plans on AMR. And it really starts to look at how the implementation of those plans are going. And in most countries, we now have the second National Action Plan on AMR. But the first one was really based on the Global Action Plan, and it was a copy and paste. By the second iteration, we're starting to see some changes within countries and some ownership in countries to actually look at what's happening. But still, we only see that 25% of countries are effectively implementing this monitoring, and only 10% are financing these national action plans. So we've still got a long way to go. And with this, we're not going to change that burden of AMR or minimise AMR unless we do that change. And we actually have to think about interventions. But I very much believe in multidisciplinary work. So we shouldn't have health systems research alone, AMR alone. We should be working together. And that's the only way we can change the burden of AMR within our human communities, animal communities and environment. That's the only way we can change that. The way that the WHO has said is, yes, we need to work together, but people need to be at the centre of these national action plans. And we need to have a patient-centred approach. In AMR, it's been a big year already. We've, certainly in the policy side, we've had the United Nations meeting. And at that United Nations meeting in September, the discussion was around having targets. And 193 world leaders actually agreed on targets for antimicrobial use and antimicrobial resistance. And those have been to reduce human deaths by 10% by 2030. So that's 6 years' time. We want to reduce the deaths. And that's based on the GRAM results. So we want to reduce the deaths by 10% based on the GRAM results. We want to implement the national action plans and to have some money to do that. Now, we might all discuss whether £100 million to implement national action plans across 193 countries is sufficient. I would say it's not. If one institute in the UK can get that much money to do research, then we need a lot more money involved with this. We want to expand the use of the access group of antibiotics. So with the number of antibiotics or the different antibiotics that we use, the access group of antibiotics are those that have the less push to resistance in bacteria. And so if we use more access antibiotics, we'll get less resistance. And that should be our go-to antibiotic as clinicians in the field. And what does that mean? That means that we should then be thinking about what we're treating. So yes, empirical therapy works, but actually if we can get diagnostics in to say what the infection is and treat that infection properly, that's where we want to be. And I would say that as a scientist, as a diagnostic scientist. So improve access to diagnostics. We want to also improve monitoring and evaluation of those national action plans. So we want to know how far we've got, what the impact is, what's happening on the ground as well. And that's through the TRACS survey. So what does this look like in practice? A year ago, just over a year ago, the European Union agreed on targets on AMR. Now, these targets are somewhat different to those agreed at the UNGA. So they agreed to reduce the total consumption of antibiotics by 20%. This wasn't agreed on by the UN high-level meeting. Agreed to use at least 65% of total consumption be access antibiotics, so a little bit less than was agreed on at the UN high-level meeting, to reduce the incidence of bloodstream infections with MRSA by 15%, and the incidence of bloodstream infections with third-generation cephalosporin-resistant E. coli by 10%, and then the total incidence of bloodstream infections with Klebpneumo that are carbapenem-resistant by 5%. So this is what the EU decided. And how have they gone? So luckily, they produced this report just a couple of weeks ago. And what you see here, I'm afraid I'm going to have to walk. Can I use the microphone? I'm not very good at standing still. Okay, yep. So what we see here is actually the consumption of antibiotics has gone up a little bit. Yes, they're meeting their target for 65% consumption of access antibiotics, and they're doing pretty well for MRSA, and even for E. coli, that are third-generation cephalosporin resistant, but carbapenem resistance has gone up in Klebpneumo, gone up just a little bit here. So these are the targets that the EU are looking at. And what does that look like in the UK? So in the UK, we're looking at different targets, obviously, and this was published last week. So what we see is antibiotics, although the consumption, total consumption was going down pre-COVID, we're actually seeing it go back up now. And that antibiotic consumption really is seen in primary healthcare settings, not in the hospitals. So that's in the community. And when we look at the aware categorisation, we see that in primary healthcare, 67% of antibiotics are access, less in secondary care, which is what you'd expect. So we're getting close to the right target for 70% access. But then when we start looking at the burden of bacteremia, we're measuring something different to the EU. And these are two high-income countries that are looking at different things. Most of my work is in global health in low-income countries. How do we think they're going to measure this? Can they measure it? What will that look like? Many of the countries in which I work don't have good quality functioning laboratories. How can they measure a lot of this? It's very difficult. And just to highlight that, this is some of the work that MAP, so the Mapping AMR and AMU Partnership, published as part of the Fleming Fund operation. And what they did, they looked at, or they went into 50,000 laboratories in, I think it's the 14 countries, in which they work. They went into those laboratories, and only 1.3% of those laboratories could actually perform antibiotic susceptibility testing. That's what happens on the ground. That's what it looks like. And then when they looked at that, they found that, yes, there were reference laboratories, but not many people could even get to those reference laboratories. So even if the laboratories worked really well, people couldn't get there. And so they went through this. They went into a bit more depth. How many of those laboratories had electronic laboratory systems? How many of them could do the ASTs and actually identify pathogens? And the picture wasn't fantastic. And Fleming Fund has been doing a fantastic job just building the capacity in countries And this, I would say, is probably the best that's possible on resistance without more capacity building, without more support. So that's on the antimicrobial resistance side. What about antimicrobial use? This, I would say, antimicrobial use often, until very recently, has not been reported, certainly not recorded electronically. So a lot of the work that MAP did was to get paper records and make that electronic. And they were able to get paper records for 1 or 2 years from 12 of the 14 countries in which they worked. And the really good news for them was that access antibiotics, actually, it was at 70%, it was even better, it was fantastic. They reached that target. But then they went into it, they delved into it. And that was because these hospitals only had 4 antibiotics available to them, and they happened to be access group of antibiotics. And very few of them actually came from the reserve category of antibiotics. And if you think of this, this is where resistance is higher. We know resistance is higher, so we need good antibiotics to treat those resistant organisms, but they don't have them. So it's the access to antibiotics that we talk about quite a bit. So now I'm going to take us back to the WHO and their people-centred approach to actually looking at antimicrobial resistance and to minimising AMR. So they do recognise there are lots of system challenges. They know about this. They can see what's happening. And they've actually had, they've talked to a lot of people and they've got a lot of information in to understand what that looks like. And there are people challenges. So people don't understand what AMR is. In many countries, they don't even have the words for antimicrobial resistance or antibiotics. It becomes a whole sentence to explain what an antibiotic is. These are some of the problems that we have in trying to minimise AMR. And I would say in working in silos, rather than working with partners in many places. I know the Liverpool School does a fantastic job of this, so I have to applaud you. So at that point, I want to sort of say, this is, that's where we are at the moment. And I want to introduce you to a study that I'm running in Uganda. A lot of what we have, as I said, is hospital-based data. I'm really, really interested in what's happening in the community. In my mind, what happens in the community is somebody gets sick, and so they go to the cupboard, the medicine cupboard in their house, or they go to a friend, or they go to a family member and they get antibiotics, or even to the medicine person in the village and they get some antibiotics that way. And then it's only when they get much sicker that they then go to a pharmacy and they get antibiotics there. And when they get really, really sick, they go to the hospital. That's how it worked in my mind after living and working in Asia and Africa for many years. But actually, when I speak to people, they go to the outpatient department. They go to the hospitals. So maybe what we see in the Graham study and what we see in the GLASS WHO work is right. Maybe we have the resistance right. But I don't think we know that. So my study in Uganda, CAST UTI, is looking at people who arrive in pharmacies in Uganda with uncomplicated UTIs and collecting that information on the causes of the UTIs to understand, is GLASS right or not? So we're going to make that comparison to start with. We also have other questions in here. We've got 2 different communities, one in an informal settlement, one in a richer area, and we're going to look at the differences there. We're also going to understand what antibiotics do people take? Why do they take those antibiotics? What antibiotics are available in the pharmacy? What can people get? So just start to look at all of that. And the other thing, so the map here just shows how many pharmacies there are. So down here, Mianga is the richer area, which has 5 pharmacies. Whereas if you look at the informal settlement, I think there are about 53 pharmacies there. So 10 times as many pharmacies. And they're small, they're tiny shops. There's not much in there, a few antibiotics and that's it. So there's a big difference between these. And we need to understand that as we go forward as well. And my final slide. And the time's gone down. So the other thing that I'm involved in is knocking out AMR. So I'm running Knocking Out AMR with the Microbiology Society. And within this, we're trying to understand AMR as a complex system and how can we unpick AMR and find solutions to minimize AMR. Just showing you here, we've been looking at therapeutics, diagnostics, surveillance and vaccines and trying to figure out, can we find pathways to minimize AMR? So if we think that we want to use therapeutics effectively against AMR, what are the barriers? Well, some of the barriers are accessible and affordable therapeutics, and what are some of the solutions? So we've got push incentives, but there are high development costs. So how can we get over those? Can we partner with people? Can we do things around this? Just to try to unpick that so that as researchers, we can say, well, I'd like to tackle that bit. I'd like to do that. Can I understand that? Can I get partners to do that? So I don't have any last minute or last conclusions here, but just to say thank you very much and open to questions later.
Thank you very much, Katrin. I forgot to say at the start that we're going to take all questions at the end after the 4 first talks. So all of the speakers will be here. So if you've got questions, please hold them until the end. Write them down so you don't forget them. Equally as important for every speaker. Or submit them online, as Fabrice is just itching to be able to field all of those questions coming in. Okay, so the next speaker we have is Patrick Musicha. Patrick, from MLW and LSTM, who's going to talk to us about evolution of AMR.
Thank you. I'll talk about transmission of AMR. It's good to come after Katrin because she's laid the foundation of the burden of AMR. One thing to talk about or to say is that the highest burden of AMR is in sub-Saharan Africa. And so we need interventions to address the spread of AMR in low and middle income countries, particularly in sub-Saharan Africa. But those interventions have to be backed up by data. And one of the widely accepted concepts is that AMR is a One Health problem. But this is not leaded, very much supported by data, especially from low and middle income countries. So I'll talk about transmission of ESBL producing Enterobacteria C and the genes across One Health compartments. So just for a start, ESBL E. coli that are resistant to third generation cephalosporins, antibiotics that are widely used in the world, but also more importantly in low and middle income countries. So the first question is AMR a One Health problem? As I said, people would say yes, but recent data from high income countries has shown that there's limited evidence to support that concept. So I highlight 2 papers here which found no linkage between human isolates and isolates from the environment or animals. And there are several other papers, and these papers have raised the question as to whether indeed AMR is a One Health problem. However, when we look at this, in low and middle income countries such as Malawi, there's a very close interactions between humans and the environment than it is in the high income countries. And this level of close interaction makes us to think that the picture that we are seeing in the high income countries might not be the same as what we would see in low and middle income countries. The DRAM study, which is the drivers of antimicrobial resistance in Uganda and Malawi, was set up taking a One Health approach, using a household as a sampling unit and collecting samples from individuals within those households, the environments close to those to the households, as well as animals kept in the or associated with the households. This study was conducted in 5 sites in Malawi, 3 and 2 in Uganda, and we undertook several sequencing approaches, including microbiome, shotgun metagenomics, whole genome sequencing, and so on and so forth. And what I am presenting to you here is results from the analysis of the genomes of ESPO-producing E. coli. One of the things that we wanted to find out is to understand the diversity of these genomes. And as you can see, we found over 170 different sequence types, which was clear that the ESPO-producing E. coli in these settings is very diverse. However, one key finding that we saw was that the sequence types were not restricted to a particular ecological niche. So you could find the same sequence types in humans, in the animals, as well as in the environment. Is this enough to tell you that that's a One Health problem? Probably not. One of the things that you would see is that so we undertook phylogenetic analysis of the genomes, and if you can see from here, some of the isolates are very closely related, some of them almost identical, as you can see from the phylogenetic trees with short branches. But you do so also see within sequence types isolates that have very long branches, indicating that they are not similar. And when we quantified the genetic distances between the different isolates in the trees, you could find that there are isolates that differ by more than 6,000 SNP distances. And from that perspective, you could actually say that probably the sequence type is not a good unit for measuring transmission. So you cannot use the sequence type as a measure of transmission. And therefore, we went ahead to undertake high-resolution SNP distances. So this is a transmission network that we constructed by connecting only genomes that differed by a maximum of 5 SNPs. And this is a very high threshold. But what you would see is that I've picked some of the examples of the clusters that we found. So in each cluster, there are isolates that are only connected by a maximum of 5 SNP distances. And what you'd see is that in each of those large clusters, you have isolates from the animals, from humans, as well as from the environment, which was a clear indication that there's transmission between these different ecological niches. And then the next thing was to try and quantify this transmission. The largest number of samples that we had were from humans, followed by the environment. But what I want you to see from here is that at each of the different SNP thresholds that we used, you would have expected a higher number of transmission events between humans and humans. But what you see is that there is high transmission that is consistent through the different SNP thresholds, higher between environment and humans than it is between humans and humans. And it means that the environment is playing a critical role in the transmission of antimicrobial resistance, either as a reservoir or as a route to transmission to human beings. And then we tried to look at the specific environmental samples. And one thing that I want to highlight here is this. So we found that household stored water was one source or one environmental source which was very much linked to humans. I think this is a critical point. It highlights the need for good hygiene, water sanitation, and hygiene practices to try and tackle antimicrobial resistance. Now, antimicrobial resistance does not only spread by the strain from person to person, it can also spread by the antimicrobial resistance genes or the antimicrobial resistance determinants themselves from one bacteria cell to another, or from one bacterial lineage to another, or bacterial species to another species. And the key vectors for that transmission are what we call plasmids. So we looked for the ESPL genes, we found 20 different ESPL genes, and we looked for the plasmids that were harboring these ESPL genes. We found that these ESPL genes were linked to 55 different plasmids. And we tried to to connect those plasmids to the genes. And one key thing that we found was that each gene, each ESPL gene had multiple plasmids that could transfer it, providing multiple pathways for transmission. It means that this gene could be transmitted to lineages, different lineages with plasmids that were compatible with many lineages. And then the next thing that we looked at is, again, to see the distribution of the plasmids on the transmission cluster. So this is the same transmission cluster that I showed, but here we've colored the clusters or the isolates based on the plasmid that they carry. And there are several things that you would see, several patterns. One is that you have transmission clusters in which all the isolates share the same plasmid. This cements our argument or our finding that those transmission clusters represent transmission of isolates between different ecological niches, because what it shows is that the isolates are not just similar at the chromosome level, but they're also similar on the plasmids that they carry. And the next pattern that you see is that you have clusters where the isolates in that cluster carry multiple plasmids. And again, this is also an indication of the multiple pathways through which the same gene can be transferred to different isolates. And then the next one is that whereby you have clusters that have no plasmid. So the plasmid, the ESPL genes, especially BLAST-CTXM15, can reside on a plasmid, but can also reside on a chromosome. Again, the different ways in which the genes themselves are spreading in this setting. And then lastly, you have plasmids, the same plasmid that you can find in the multiple clusters, allowing one plasmid to secrete to different clusters, spreading the same gene. So putting this all together, what we have found is that we have found highly similar ESPL E. coli strains and determinants that are shared and transmitted between One Health compartments. And the success of the diversity of the sequence types and the plasmids that carry these genes are allowing successful dissemination of the ESPL genes into multiple lineages and providing multiple pathways for transmission. When we think of all these data together, I think we can conclude that antimicrobial resistance in low and middle income countries is a One Health problem. Thank you.
Thank you very much, Patrick. It's a very convincing argument there. I'm sure there'll be questions for you at the end. It's my pleasure next to introduce Dr. Sabrina Moyo. She shares appointments across LSTM, University of Bergen, and Muhimbili University in Tanzania. And she's going to talk to us about a particular intervention which she ran recently.
Thank you, and thank you, the organizers, for this opportunity. So I'm going to give a talk on this clinical trial which we conducted in Tanzania, looking at probiotic protection against carriage of AMR bacteria in newborn babies. So thank you again, the two previous presenters, for giving an introduction of AMR and ESBLs. As it is known that WHO has declared AMR as a public health threat, and of particular concern are the extended spectrum beta-lactamases producing bacteria, because these can hydrolyze Monobacterium cephalosporins and usually they are co-resistant to other antimicrobial agents. So carriage with ESBL-producing bacteria, this is intestinal carriage, poses a potential risk of transmission and infection within the individual. And ESBL-producing bacteria are top priority pathogen by the WHO. So what is the situation in Tanzania? One of my colleagues started a bloodstream infection study in 2000, of which he found that majority of children are infected with gram-negative bacteria, which are ESBLs. But at that time, it was less than 20%. The additional thing is that mortality rate was high for those children infected with ESBL-producing bacteria if you compare with children infected with susceptible bacteria or they have malaria. And then during my PhD, I screened the children with looking at if they are carrying ESBLs, children less than 2 years. And in one of the publications, we found that very young infants, 0 to 3 months, are the ones carrying the highest burden of ESBLs, 2/3 of those neonates or infants. So I conducted a follow-up bloodstream infection study in 2017, which is 16 years since the first bloodstream infection study in the same region and the same hospitals. And at this time, I found that these children are infected with ESBL-producing bacteria, and it is over 50% of them. And still, mortality was high associated with ESBL-producing bacteria. And of those who died, 73% were neonates. These are babies 0 to 28 days. So, and from that same study, because we knew that previously that majority of infants are carrying ESBLs, so in the second fever study, we included both blood and fecal culture to screen for ESBLs. And what we found is that the same bacteria, ESBL bacteria, which was found in blood, is the same bacteria these babies were carrying in their gut, ESBL. So that proves that colonization rates increases the chances of transmission to infection. And this high ESBL colonization have now evolved towards global pandemic, with developing countries being mostly affected. So after establishing this truth of AMR burden in Tanzania, we thought of how are we going to contribute in reducing AMR infections. That's when we came up with the use of probiotics. So probiotics are live bacteria with beneficial properties. They may enhance colonization resistance and build resilience against adverse environmental factors. At that time in 2017, there was a large clinical trial conducted in India, and a randomized clinical trial with over 4,000 infants. And in this study, they used a single strain of probiotic, which is lactobacillus, mixed with prebiotic. And babies were given the first week of life, and they were followed up for 60 days, and they showed that there was a reduction of incidences of sepsis and death by 40% during the first 6 days. Supplementation of probiotics in newborns have been studied in a large number of clinical trials, but most of which are in high-income countries. And in most of those studies, they were done in preterm babies. So it is in some of the European countries a routine care to give probiotics for preterm babies to prevent sepsis, necrotizing enterocolitis, and other things. So we designed this study, use of probiotics to reduce the infection, death, and prevention of ESBL-carrying bacteria for newborn babies in the northern part of Tanzania, which is Haydom, and the surrounding areas. And this was a randomized clinical trial, with the primary objective to assess whether a 4-week course of probiotic therapy could reduce death and hospitalization up to 6 months of age in these newborn babies. And secondary objectives were to look at colonization of ESBL bacteria, taxonomy of gut microbiota, infant growth, gut inflammatory markers, as well as adverse events associated with the use of probiotics. So that is the probiotic that we used. It is Labinic. It contains 3 strains of bacteria. One is Lactobacillus and 2 strains of Bifidobacterium. Manufactured in UK, but the industry is in South Africa. So this is the hospital where we conducted the study. And this is Tanzania in East Africa, and this is the northern part of Tanzania. While most of our studies were done in Dar es Salaam, these were mostly cross-sectional studies or cohort studies, but this was a good site for a clinical trial, which is kind of rural region. So we did this randomized clinical trial with 1-to-1 ratio. We included the babies born with over 2 kilograms. Either they were born at home or at hospitals, and they either received 5 drops of probiotics or 5 drops of placebo. And both bottles of probiotic and placebo were identical, and they were administered by caregivers at home. So babies were recruited in their first 3 days. They have to start probiotics, not beyond that. So mothers were included in the study in their last trimester of pregnancy. We followed up these babies for 6 months. We had the first visit at 1 week to see how they are going on with the use of probiotic. And then again, we visited again at 6 weeks and 6 months. These are the times where we recorded if there were adverse events at home or if they were hospitalized. And we also collected stool samples for different analysis. So the study was supposed to start in 2020, but because of pandemic, it was delayed. So it started in 2022 and ended January 2023. And we were able to enroll 2,000 newborn babies with 1,000 placebo and 1,000 in intervention. And follow-up was up to 97% retention rate. And we were able to get stool samples from 96% of all participants. So these are some of the findings. From 4,000 samples, we were able to collect over 1,000 ESBL strains using culture. As you can see from the diagram, at 6 weeks, there was a reduction in ESBL colonization compared to placebo group, but the effect was not seen at 6 months period. And in this one, so as I mentioned earlier, that the effect was seen at a period of 6 weeks, but not 6 months. But that is the important period because most of the infants are having these infections and death in their first month of life. So 6 weeks protection is good enough for us. And then additional things that we saw is that babies, female babies and babies who were born in the hospital were also protected against ESBL carriage, both at 6 weeks and at 6 months. And at 6 weeks alone, those babies who were born by vaginal route and who did not use antibiotics were also protected from ESBL carriage. But one surprising thing was children born to HIV positive mothers had increased the risk of carrying ESBLs if they have taken the probiotic. So our conclusion is that we were able to reduce ESBL carriage at 6 weeks, and probiotics may become part of future strategies to combat the spread of AMR and AMR-related infections. But further studies are needed to confirm before we come up with guidelines for clinical care to give probiotics. And we did not observe any short-term safety concerns with the use of probiotics in our infants. So the work is still ongoing. We have one of our PhD students here working on looking at resistant genes and co-locations for these isolates, and one of Emre's student at LSTM who was looking at persistent colonization for those children who had ESBL at 6 weeks and 6 months. What are the factors that are responsible for continuous carriage of these ESBLs, host factors or bacterial pathogen factors? We are also still analyzing microbiota composition. So this is just the initial analysis. At least we could see at 6 weeks, there are relative abundance of higher bifidobacterium strains, which we gave to these children in placebo group compared to in a probiotic group compared to placebo. And we are still working on looking at gut inflammatory markers, looking at fecal calprotectin, still performing whole genome sequencing for all over 1,000 ESBL bacteria, and we are currently comparing culture versus direct qPCR for detection of these ESBLs to see if we have lost or we did not detect some of them. And with that, I would like to thank all the participants, the collaborators, and the funding agencies for this trial. Thank you.
Thank you, Sabrina. It's a very promising area of work. Hopefully we'll hear about future results soon. So our final offered talk in this session is from Tony Nolan, and we're going to change tack and look at resistance in vectors and how we can possibly mitigate it.
Thank you, Adam. Nod to the AV guys at the back. This setup is lovely, so thank you very much. So if you're wondering what I'm doing here as a mosquito gene jockey in a session on antimicrobial resistance, exactly, thank you. You stole my punchline. Adam asked me basically to talk about gene drives and resistance, and if I may say so, that was an inspired choice, not because of the quality of my talk in any way, but because there are many themes that unite the dynamics of resistance, how it's selected, how it arises, and how we can actually combat it across different interventions. So today, I'm going to talk to you about gene drives and some of the potential benefits, but also some of the challenges, among which are resistance for gene drives. So in the interest of time, you're going to just have to take my word for it that we and others here at the school have developed a whole range of different genetic modification techniques that allow us to understand some key traits that make mosquito vectors such good vectors of the malaria parasite. That can either be an intrinsic susceptibility to the parasite and disrupting that, it can be susceptibility to insecticide or resistance to insecticide, or it can be other behavioral changes or impairments of reproduction that potentially offer routes to interfere with parasite transmission overall. And this is the concept of genetic control. So imagine we have a modified mosquito, just take our word for it, we can make these. How do we actually make that into a meaningful intervention in the field? And there is the challenge, okay? Because suppose we have a modified mosquito, if we release that into a wild-type population, the frequency or the percentage of modified mosquitoes in the population in the original release generation is only a tiny fraction, and that is not expected to change generation upon generation, because any single genetic trait is only inherited by 50% of the offspring. And so that's a problem, also because the modifications that we introduce are not likely to increase mosquito fitness, and therefore the initial release frequency will remain low over time. And that's where this concept of a gene drive is useful, because in all likelihood, these modifications are going to be lost from the population over time. And so a gene drive, compared to normal Mendelian inheritance, where this genetic modification is eventually diluted in the population, a gene drive is a biased mechanism of inheritance whereby a particular trait is inherited to all the offspring. And therefore, with each generation, it increases in frequency until it can actually go to fixation in the population. So the definition of a gene drive is any genetic element that's able to bias its own inheritance, and it can often do that even if it's spreading with a fitness cost. Gene drives, such as many selfish genetic elements, exist in nature, And we've taken inspiration from one class of gene drive, which is called the homing endonuclease gene, to build our own synthetic versions of these genes. And these things work in organisms that have 2 copies of every chromosome. You have a genetic element that is designed. I'll use this pointer, otherwise people at home can't see it. No, I won't. I'll take back what I said about the AV guys. Okay, so this is a very simple design. You have, has anyone heard of CRISPR-Cas9? Some of you will have heard of this. This is just a very specific DNA cutting enzyme. And it's designed to recognize on the wild-type chromosome a very specific target sequence, which is specified by the provision of this guide RNA. So you have a genetic element that's inserted on one chromosome. By production of this Cas9, we get cleavage of the alternative chromosome, and this gap has to be repaired by the cellular repair machinery, and often to do that, it will use this as a template to fill in the missing sequence. So what that means is this genetic element, from having been on only one of the 2 chromosomes, is now, after the repair process, on both chromosomes. And that's how it increases its copy in the genome. Okay, so this is a very important phenomenon, and we decided to see whether we could recreate that in the mosquito. So the basic idea is to insert one of those selfish genetic elements on one chromosome and see if it's able to copy across onto the other chromosome. And if it can do that in the germline, which are the cells that go on to produce the offspring, then all of the offspring would receive a copy of the genetic element, regardless of whether they inherit this chromosome or this chromosome. So that's the basic principle. And so coupled with this genetic element, we can either introduce traits that negatively affect the population and cause its suppression, or we can introduce traits that modify the population because those genes are somehow acting against the parasite and making those mosquitoes intrinsically resistant to the parasite. The approach that we've used is to spread genes or mutations that are detrimental to the mosquito population and thereby suppressing it as the selfish genetic element accumulates in the population. And so, in principle, what we have is initial release of a gene drive at low frequency in the population that has this selfish genetic element. Because it's making a recessive lethal mutation, individuals that harbor the gene drive are absolutely fine because the gene is recessive. It accumulates in the population over time silently until it reaches such a frequency that we start to get individuals that have both copies of this recessive lethal mutation. And in those individuals, that lethal mutation makes them non-viable. And so that leads to population suppression because these are non-viable. And so as the gene drive is increasing in frequency, the overall fitness of the population is drastically reduced. And so take it from me that we can build these. When we build them, they pose a lot of potential questions, one of which, for the purposes of this session, is resistance. But also there are ecological and biodiversity effects if we're considering suppressing a population with a selfish genetic element. There's the issue of containment and how to get community and regulatory acceptance for such a type of intervention. So if we talk about resistance, which is the reason why I was invited to this session, for gene drives that have a very specific recognition sequence like this, the most obvious source of resistance is going to be variation at the target site, which renders the gene drive no longer able to recognize it and insert within that allele. So if these have increased fitness in the presence of the gene drive, we would expect them to be selected in the population and the frequency of this allele to increase over time, possibly go to fixation. And so in the first generation of gene drives that we built, while we showed that they could initially increase in frequency despite the fact that they're having a very strong fitness effect on the population, over time as we continued the experiment, their frequency in the population started to decrease away from the modeling predictions. And we imagined that this was due to the emergence of resistance. And so we tested that simply by looking at our most preferred hypothesis, which was that there would be variation at the target site, and sequencing the populations at an early generation in that gene drive invasion and at a later generation and see if there were changes in allele frequencies. And indeed, what we observed were small in-frame deletions that were initially at low frequency that started to be selected in the population because they are conferring resistance to the gene drive while having minimal fitness effect in the mosquito. So as I said at the start, that means that gene drives are no exception. They're not a magic bullet. As with all technologies that look to suppress a population, be it antibiotics or insecticides, or even vaccines to some degree, they can all select for resistance, and resistance selection is to be expected. But just as with these, the gains to public health are not in question, even though we can select for resistance. The same is also true for gene drives, essentially. And with gene drives, because I've just shown you the most likely form of resistance, we can say that it is, to some degree, foreseeable, and various strategies exist to reduce the likelihood of resistance arising in advance. And so here, I just want to show you that this idea, this concept that some of those considerations when we consider resistance are shared across insecticide use, across antibiotic use, and also gene drive. So standing variation in the population is important. The fitness cost of the resistance allele will drastically affect its rate of spread. This one perhaps less so, but also to a degree. The extent of reproductive isolation matters in terms of how fast that resistance mutation is likely to spread. But this one, in terms of mitigation, is also shared, and that is this concept of targeting functionally constrained sites, because these are less free to mutate and confer resistance. And the same is true for gene drives. So if we go back, redesign our gene drives to target very functionally constrained sites in the genome, we can release those into a population. They can go to fixation, and as they go to fixation, because they're spreading these recessive lethal mutations, eventually the population is non-viable and you don't get any offspring, and the whole population crashes. We can repeat that at scale in semi-contained environments that are more ecologically challenging for the mosquito, and have those experiments run over longer time periods, and we see exactly the same phenotypes. The drive goes to fixation in all replicates and crashes the population over time. And so it's these types of things that allow us to make inferences about how a gene drive might behave in the field with those more ecologically realistic settings. And we can see that, for example, this is a map of Burkina. This is modeling. It's theoretical, I should add. We haven't released the gene drive yet. And we can see that the gene drive, if we release it in only 1% of settlements on a single release, over 4 years, we expect extensive spread. And we're going to see areas of very high suppression and areas of more intermediate suppression. So the message here is that the gene drive is invasive. It can have large-scale population suppressive effects, but these are heterogeneous. And this is important to remember. And it goes to that theme that we heard yesterday, that any intervention, even gene drives, has to be considered as part of a wider approach that is multi-combinatorial and using a range of different vector control and parasite control approaches. So with that 10-minute talk on gene drive, which is challenging, I admit, hopefully I've given you a flavor that they are self-sustaining, species-specific tool with potential for malaria control. Both population suppression and modification strategies are available. But as with any other technology, managing resistance is going to be key for robust outcomes. And what I didn't have time to talk about is that gaining community acceptance is going to be crucial for success. I'd like to thank everyone that's done the work on here, but I don't have time, and Adam would kill me probably. But just to point out that this was work that was started at Imperial College, continued here with my group, and in collaboration with IRSS in Burkina Faso. Thank you.
Thank you, Tony. That was fascinating.